Verónica Dumit. Thank you again, Verónica, for being here, and thank you for these two amazing talks. I haven't seen the next one, but I'm sure it's going to be amazing. So thank you again, and all yours.
Yeah, thank you, Herman. Thank you, Nico. Good morning, everybody. I'm going to continue with the talk from yesterday. I start with what I showed already, but just to remind you that for me, lung, it's important. So what I'm going to show, it has a strong focus on lung. And the reason being, it's because I'm interested in the risk of the nanomaterials, what they could produce when they are inhaled, so that the exposure for humans be in the lungs. And yeah, as I mentioned yesterday, this is the way that for toxicology, the full mechanism of a substance causing fibrosis can be described. And you remember that we talked about key events that are always part of this adverse outcome pathway. And inflammation is one of these key events leading to fibrosis. That's also a reason why you will hear that I talk about inflammation of the lung. Yeah? And well, about the omics and proteomics, I also mentioned why I'm interested in proteomics. For transcriptomics, it is already a lot done, and it will already be soon for risk assessment. But transcriptomics, it's not so close to the phenotype or to the adverse outcome. And proteomics, it's closer to the phenotype. The problem is that it's not so standardized. How you prepare your samples, how you measure them, which machines you have, the output you get, this is all very different. And then the analysis, how do we proceed with the analysis? The standardization, it's key here, so that it can be useful for risk assessment and for regulatory purposes. And yeah, I already mentioned that the first thing I did was to look what is known about nanomaterials from the proteomics perspective. And there are some projects, but not too many, and the data sets that I can find, they are not enough to go further into the mechanism or the mode of action of the nanomaterials. And that's why we decided to go to the Pride Archive, that's a proteomics repository, and to, among these 16,000 projects, to look for those that belong to lung projects. Alterations can be disease, or can be substances, or can be COVID, fibrosis, or any of these effects that I could collect from the Pride Archive, we kept. And we call this the lung proteome universe. Yeah? And we developed a workflow, that's what I'm going to talk today, how from each one of these projects we can get descriptors. And yeah, and then we process the same way the projects that belong to the nanomaterial part, to those projects, and also to data that we generate ourselves. And this is what we call the nanoproteome universe. Yeah? And for them, we will also have descriptors. The idea is how then we match the descriptors that we find for the nanomaterials into this lung proteome universe. And yeah, for this, it's key to go into data analysis. And this is a field for artificial intelligence and machine learning. And if you have a bioinformatician in your group, you can only profit from that, from the data and from how to interpret it, how to read it, how to use it. And yeah, this is an opinion paper that I read a while ago, and I recommend to you. This was written by Paul Nurse, and he thinks that biologists must generate ideas as well as data. So coming up with ideas, it's even more important in producing data. And the data can give you these ideas. You have to know how to approach it. Oh, I would like to mention the concept of fair data. This concept, the first time it was mentioned was in 2016. It was, this paper was published in Scientific Data. And scientific data has an impact factor of 6, so it's not too high, but this was a real important concept. And many things changed regarding data since then. So the people, the authors, they are from different fields, what we call stakeholders. They are from the industry, or also from the university, and agencies that also deal with risk assessment or with just health. So they are from different fields, but they all coincide in that we need that the data that it's produced has to be fair. And fair in this sense, it means that it's findable, accessible, interoperable, and reusable. And that everything that is up there, that it has to be useful or machine readable. That if you have a script or if you have a code how to approach that, it has to produce something. You can read it, you could be able to read it, and also to make your own conclusions. And for this, what they propose also here, it's that you need an infrastructure. It's not that any one of us producing, we can upload to Google Doc or to your own server, that it's not going to work. So infrastructure of data, it's important. And the format of the data and the metadata. And since then, since this time, most of the grants that the projects get, when there's a consortium researching, let's say in my case, nanomaterials, and we get millions from the European Commission to investigate that, the condition is that everything that you publish, it has to be in an open source journal, but also that your data will have to be fair. And this is becoming more and more important. And I mentioned this because maybe you did not hear about that yet, or maybe you're just starting to hear, but this is also what will come. And my advice to you would be, if you have any project, whatever the topic, you should first start by going to internet and trying to see what is there in the repositories, in databases, in such infrastructure. Because if you find it there, then you can reuse that. And as a starting point, it's really good. So when, I come back to what I said I would do, I try to have these descriptors for nanomaterials and descriptors for lung alterations. So the first thing we did was to look for a repository where we could find this data. And among the many that are there worldwide, we consider PRIDE the one that we liked to start with. And there in the PRIDE, we found many projects involving nanomaterials and the effects, but they were not known to the nano community. And they were uploaded, you could find the results, you could find the max quant output and the raw data, but no one knew in the community that these projects were done, that you could get the information. So our first task in my group was to try to verify this data, because everything that is in the PRIDE or repositories, it's not really fair. You have the things, you have the files, but you don't have the metadata that it's needed. You cannot just run a script on them, and that's why to have this possibility of fair data, you have to verify the data. And this is what we did for these projects. We brought them, or we linked to the eNanoMapper. This is a nanomaterial database. So everything that is produced in these European projects has to be uploaded to the eNanoMapper. And we linked all these new projects so that we could find in the PRIDE to here. And this means to put the metadata, to investigate into more detail what was about the project, and then explain in the format that this database needs. And now all the nanomaterial community can also profit from these projects. That were not too many, maybe there were 10, but they will become more in the future. So this is an effort worth doing. And yeah, we linked these projects, but still we want to profit from them. And also all the other many related to the lung. And for this we needed a protocol or a workflow that can process all these many projects in a time frame that we can still manage. So we of course can use Perseus, and we can try to manually to extract all the information, but if we want to process 200 projects or 500 of them, it's not going to work. So we came up with a workflow that starts with a query in PRIDE, how to retrieve the information that you want, and yeah, how to process that, considering that with the data you can run MaxQuant. Sometimes you have some raw files that are not, you cannot run MaxQuant with them, so this we decided to leave out. But still there are many. This was not a limitation for how many projects we could find. And then the workflow produces an output files, and the one that we care the most are these descriptors. But we also produce some files, or the workflow produces some files related to judge the quality of the data set. That's also important to know. So here it will be the first part of our workflow. I separated in different parts because the full picture is very long. But the workflow runs automatically, and yeah, you have your protein groups, that's the file that you need, and it will give you the output automatically with some little help from you. But yeah, we start here with the search and the PRIDE. And it's not really, yeah, it's okay, the search, but if you want to have some term to exclude, or if you want to look for simultaneous keywords, sometimes it gets complicated. And also if you want to produce a list of all the projects that you are interested in, for example, in my case, if I look for lung and I find 500 things, it's very difficult to have that in a format that then you can use. So our workflow can optimize the search, and also will give you a list, a text file that you can know all the projects that you have and what is the information related to them. Is it label free, is it TMT, which machine, which device it was used, and so on. And then it makes what you always make with Perseus, so you take out the contaminants, everything that was identified by one site. If you want to take out everything that was identified by only one peptide, the workflow will exclude, will transform the data in the log2, and this is all done automatically. But still there are many challenges when you have proteomics data, and especially when it's public. You just got this from the PRIDE, and in many or the most of the project you are not able to know what it's what. You see the samples are called like this, but what is the control? What is the treatment? How many replicates you have? Do you have different treatment, one control? Well, you never know. You're never sure. And so this is the first challenge. The second challenge is to detect outliers. Sometimes you can see this one, it's an outlier. You just from looking at the table of the intensities, you say, this one looks very different. but it's often the case that you cannot see it, but still an outlier will affect your results very much. So the next steps in the workflow deal with that. And here it's important, do you know the sample names? Do you know, is it clear from what you have, where is the control, how many replicates, how many treatments, do you know that? If you know that, then you looked for the outliers. I'm going to show how we do this. And if not, you still have to check for outliers. But then, the question would be, do you know at least what's about? Do you have how many treatments are there? Do you know that or still don't know that? So this is the minimal information that you can add. It will always help if you do, that you tell the workflow these details, but still, if you don't know, the workflow could operate on them. And I don't know R, I cannot use it. That's why to develop the workflow, a bioinformatician was, she was the one who developed everything. And for this, we use a package that you can see there. Maybe if you're familiar with that, you will know what I'm talking about. So here we have PCA, and the plot it's right 3 dimensions here. And this is what you see in 2 dimensions. This is the classic principal component analysis that you all know. And here if you do this with Perseus, that you have to manually check for what is an outlier. And then you say, okay, in the PCA, I see this sample, it's very far, I will consider it an outlier. In this workflow, it's automatically done. It's quantified also what it's an outlier. If it makes a threshold, it will be considered like, okay, still a sample, and if not, it will be excluded. And for this, we use this package, as I said. And then you took out the outliers, and the workflow will try to find out clusters. If you don't know how many clusters you have, it will try still to arrange all the samples that you have into clusters. So here, in this case, we know that there are 3 different things. It's 1 control and 2 treatments. But the workflow tells us that it's 2 groups. And that's why what you see in red will include more samples. But in this case, one of the treatments that are evaluating, actually it does nothing. So it's very similar to the control. And that's for the workflow what we are going to consider. We have 1 treatment that really has an effect and another one that not. So for the workflow, it will be like 1 control and 1 treatment. This is another way to, this method here, it's a k-means to look for the clusters. We also included this nbclust. It's another option, and we compare both methods trying to look for how many clusters we should have. So here, the important thing is when you see a huge gap. This is automatically done, and you say, okay, the gap, the highest gap is here, so I'm dealing with 2 clusters. That's what the workflow sees, and that's why it will classify it like that, in 2 clusters. Well, then you tried. So now you know with the clusters what you will consider treatment 1 and treatment 2, or according to how many clusters you have, how many treatment the workflow will think you have. Then, well, you take the outliers, that it's important, and you have to filter according to how many values you have. And the threshold, we tried different cuts and different thresholds, but what we decided is to use that we keep all the proteins that are represented by 70%, meaning that in that cluster, we know for a protein, at least in 70% of them. in one group, that it's enough that appears in one group. And yeah, but still, after you filter that, you will still have many missing values, as yeah, it happens with proteomics data. And for this, we also do an imputation. I don't know if it's, yeah, so we impute these values with another normal distribution that it's shrunk and downshifted. But this is exactly the same as Perseus does. We could find an air packages that does the same, and we could include it automatically in the workflow. You can do the same with Perseus. And yeah, and then what we have to do, just we have the data, we have now no missing values, so it is about now looking which proteins are altered. Sometimes if you don't know what is the control and what is the treatment, you are not able to know if your protein of interest, what is significantly altered, if it is upregulated or downregulated. But that doesn't matter. You still have that protein that it's altered and it's regulated. And that's why here I have a set of around 20 projects that I'm going to show. Here we make a cut for how many proteins we consider to be significantly altered and how many KEGG pathways are altered. This you can add these terms as we saw before, how you are used to that. The workflow can also implement this automatically. You can also add the Go terms or reactome, and if you have any other that you can consider to be a descriptor, the workflow can include that. And here, among these 20 projects, we had several cancer ones, and we wanted to see how they cluster. So we had cancer, everything that you see red here, it's cancer projects, and the other ones are other things. Some nanomaterials, some COVID or influenza, or some bacterial infection, fungal infection, and so on. And you can see that if you then sort them according to the KEGG pathways and how they are altered, these KEGG pathways, you see that the cluster projects, they all fit together. And for this, it's projects that we just know it's about cancer, but they were completely different and done by different groups and different years, different machines, and still they cluster together. Meaning that if you have a workflow to process the data like I don't think, I just do, you will still find things that resemble each other, and that they are still completely different from other treatments that are different. And if you focus here on this cluster only in the upper part, you see that it even works better. That then the cancer projects, they cluster all together. Here, this one, it's also a cancer. If you consider all the KEGG pathways that are available, I don't know if it's 300 or something like that. But if you focus here in the upper cluster, it works even better. This is the full workflow. So all the parts are here. And this is part of the nanoinformatics project. This is an EU project. And it's about making models to assess the nanomaterial safety. And it has a platform where all these models will be available. And that's why this workflow will be part of the platform, and we will also upload it probably in January. And if you would like to have it, of course, you can contact me and I can give it to you. Now, as I mentioned before, I care a lot about inflammation. And what we're working right now, it's how to describe inflammation. Because if you go to the right and you type inflammation, you get anything. And because inflammation, it's not like endpoint, or it's not an adverse outcome by itself, it's very difficult to know why it happens. It's the cause of something and leads to something else. So how do you describe inflammation? And for this, our approach was a literature review to go to see what is out there. And we gather, but not from the proteomics side. Here we took everything that was published and we tried to make our own inflammation map, or at least like a small set of information. We have some genes and pathways that are altered, but we're still, what is the cause and what is the effect of inflammation, it's hard. So, but we're still working on that. And we could find some KEG pathways that are involved or related to inflammation. And the project that I showed before, we said, okay, if this is the list of inflammation KEG pathways that we could come up, here we see also how our projects can cluster together. And that's we hope that it will give us more information for the nanomaterials. But this is still a work in progress. That's how we plan then to link according to which are the pathways that are altered, how you can connect the different projects. And that would be the end of my talk. I want to introduce you to my group. This is where I work. This is in winter and this is in summer. And Andrea Hasse, she is the head. And Adeline, she's only here. She is the bioinformatician that put all these tools together into the workflow. And if you have any question for more specific for the bioinformatics and how we process the data automatically, I can always contact you with her. She could give you more details that maybe I'm not able today. I thank you very much, and of course to Nico and Germán again, thank you.
Thank you, Verónica. That was so interesting, really. I want to ask you something about the computing, but just the hardware that you use to process that. Do you need a cluster for that?
No, it's a computer. We bought it new from the 2 years ago. The group bought it and it cost 2,000 euro.
Okay.
Now it's something affordable, and it's a normal computer.
A normal personal computer. Okay. And did you train your algorithm, or did you test it with in a biased way? Have you tested if your algorithm makes mistakes, or did you train your algorithm in some way?
We have many data sets that are obviously bad. When you look at them, you obviously see that the quality of them it's really bad. And what should be a replicate that's not correlate, and you see this is really a bad quality project. And if you process it with a workflow, you get nothing. That's why we are confident that if you have something that doesn't make sense, you are not able to get any of the information. But we never had the case that we suspect that what we got was wrong. Because it also, for those projects that there is a publication, it's not always the case, but for most of them you have a publication. When you go to the publication, it fits. What we've found here with the workflow, it's similar to what they claim. Sometimes it's not to the very last detail, but still we could never have one case that we could say, no, the results that we got are bad, are wrong.
Great.
But
Thanks.
sometimes, and it happens that when you have, as for example, you can see here in this 20, the group of 20. So these projects that we have for this set, when you cluster them, you don't have as many here. It's because the missing ones were empty. They showed that nothing was significant. Or yes, some proteins were significant, but not the descriptors. And then, well, they are lost. But we believe that the strong part of this is that you will be able to have very many projects. And then to retrieve the information from the majority.
Any question from Verónica? Jon over there.
Thanks a lot, Vero. I think you really nicely showed how you can mine all the data that is already out there and generate new knowledge. So now from an academic point of view, because you are now at the research institute, at the Federal Research Institute, right? So do you then also plan to publish that? Because that would be like a bioinformatics paper that could be published, or because this is now part of this EU project, this will just go into this database? How is that?
No, this we plan to publish. We have a manuscript, and maybe now in January we can submit for the workflow. We would like to just present that it will be also enough for the nanoinformatics project for what we aim to do. It's a model how to handle this data. And what we would like to publish, but that will have more scientific relevance then, it's this part with the inflammation. If we can then cluster our nanomaterials to some other lung alterations that give us a bit more information into the mechanism. But the workflow, yeah, we're confident that we can present.
Yeah. A second question. I think it would be also very much in the interest of Pride, for example, that their data can be reused and that you link this to this nanomaterials database. So did you contact the people in Pride for this workflow, or you just basically used their interface and that was it?
No, we did not contact them, and we just, this linkage that we did to the eNanoMapper, it was the description of the metadata and where it is located, because the database for nanomaterials cannot host such amount of data. It's many gigas, and that's why the only important thing is to make the link, where you find it. And no, we did not think of contacting Pride because we use it as a normal user.
Okay.
So far.
Yeah. Thank you.
Thanks.
Vero, this algorithm, have you developed it from scratch, or there are previous examples of something similar of what you are doing?
Well, we started with MaxQuant and Perseus analysis the way I knew, so the way you know, and the classic way, how to approach proteomics data. But then you have to do it manually, and you have to sort the outliers manually, and everything. Then it needs your input, your constant input, and that's why we decide now it's not going to work like this. We need something, and also standardize. We want to go further from the bias that we can bring to the analysis. But basically, it's everything that you find in Perseus. But here you have an ERB package that will quantify the outliers, the cluster. When you impute the values, you will also have, you had a normal distribution, and then you will have an additional normal distribution. The resulting distribution is still normal, it's still acceptable. Do you affect the results with your imputation? And this is something that in the workflow we can quantify, and then in the output file you get like a quality check. Things that went okay and things that then maybe distorted the things a little bit.
Right.
But basically you could do the same. If you know Perseus, you can get the information.
Questions for Verónica? Yes, Juan. then Eduardo.
Hello. I wanted to ask you if at the end of this you are hoping to have like a list of descriptors for each of the situations you are evaluating, and then with that list of descriptors, are you hoping to see if you can look for that in vitro assays, or somebody else can look for that in vitro assays instead of be using animal models and stuff like that?
Yes.
I mean, how this connects to the first part. I was wondering how this will reduce.
Exactly. So if I go back. Here. So these descriptors from the lung, what I get here that can be the protein ID, GO terms, and GEG pathways, and so on. I want to see how the ones coming from nanomaterial projects fit to there. And we can also produce new data here. And we can produce new data, let's say just in vitro, you have cells in culture and you apply the nanomaterials and then you see the effect. This we can do. And we can also try to use, let's say, the nanomaterials resemble to this substance, to do the experiments ourselves and then confirm that the effect was similar. But actually, it's what we wanted, just to match these descriptors into anything. Let's say it looks like influenza, the effect. Because of all of them, of all these projects, we know much more than what we know for nanomaterials. So maybe just this linkage could bring us a bit into the direction what we should look for. Because then we know maybe influenza causes that. Okay, then we can do another experiment, not proteomics, but knowing what we want to look for. That would be our overarching aim here.
Thanks, Verónica, for your talk. It was really nice. And I'm wondering that you are connecting two big words like nanomaterials, that is so diverse, and proteomics, in some cases related to some kind of symptoms that are so diverse, like inflammation. So I'm wondering if you, if that can give many false heats that you need to purify in some way, going one by one later on. I see like a huge amount of work, and still you will have so many maybe false positives.
Yes, we wonder that ourselves too. Yeah, if it's going to bring us to the right track. But I have many reasons why to continue this. Because when you think you have an answer, let's say, what nanomaterials or those nanomaterials do to cells, or what they should do to the lungs, it's very similar to influenza. We are not going to make that conclusion. Then you know what to look for. Then you can have additional experiments to prove that. And so that it's not going to be the end. That's going to be just the start, how we can orient ourselves what to look for. And so that's one thing, that to give us the direction, what it might be that we should pay attention to. And also because I believe that data, it's the future. It's the new commodity. You have data, you own something with great value.
Yeah.
And to try to start now thinking of that this is important, that also it will become more important, the amount of data that it will be out there, the power of your computer, the cost will be lower, and the future it's that. We have done so much in science, especially in system biology. We know so much, we produced so much data. How can we use it? I don't think we extracted all the information that we could.
Yeah, I agree.
And that's why it's my personal feeling or my opinion that we have to start trying to profit from data.
Yeah.
It has no cost, or only minimal if you compare to working in the lab. It has no cost. I don't know, the computer we have and we use all the time cost like, I don't know, 8 antibodies. And with this we already been trying to make infrastructure of data for a long time and trying to use that. So that's why it's my personal wish to go into this direction. But yeah, it's still will be a challenge to see does this make sense or not.
Thank you very much. That is important also to know that we should make tools to get the data readily available and to improve the quality of how the data is saved and the clarity and the quality of the data, because sometimes the data is lost because it's not properly annotated. Or
yes, that's why we try to cope with those challenges. But I don't know, when I started Proteomics 10 years ago, you could publish your results. They did Proteomics, you could publish. But then, in the meanwhile, you cannot publish if you don't upload the results. And it will be that then now you have the results, but it's not clear what was there. But the newest projects, the metadata, it's more complete. You have even the outputs of the MaxQuant or whatever software you use, but it's also uploaded. And I believe that in the future this will be more clear. And I think, and it's what I see, that you are not going to be founded to have a grant if you don't comply to these rules, that you have to bring the data into this structure. And yeah, I think it has future. But it's my opinion. Welcome.
Questions? Oh, no, another question. Come on. Okay, now you can upload. Vero. Thank you so much for being here with us this week. Verónica is going to stay until the end of the course, so please reach out to her if you have any further questions. Vero, thank you so much. This is for you.
Thank you.
Thank you.
Thank you all, and yeah, thank you very much. Nico.
So we are going to have our coffee break right now. Please, those people that are going to have their flash talks for today, you can send the files to Rodrigo over here, and we'll meet in 20 minutes. All right.